Virology invented the fictional theoretical virus model, but has consistently failed to fulfill its own requirements to prove their existence. It is claimed that viruses cause disease after transmitting between hosts such as humans, and yet, outside of cartoons, computer models, and Hollywood movies, the scientific evidence for these claims is missing. One of virology's greatest failures has been the inability to obtain any viral particles directly from the tissues or fluids of organisms like humans said to have viral diseases. In order to obfuscate this state of affairs, virologists have resorted to creating their own pseudo-scientific methods to replace the long-standing scientific method, like rigorous control experiments, to try and disprove their original hypothesis. But virologists have also nefariously changed the dictionary meaning of words in order to mislead the public and support their anti-scientific practices. For instance, to isolate means to cause to be alone or apart from everything else. Yet virologists have never isolated this extracellular vesicle particle alone and apart from the thousands of other particles found in human tissue, fluids, and their petri dish and test tubes full of monkey kidney and fetal bovine calf tissue, to genetically characterize it and prove it even is a virus particle, and not just the usual, normal, regular dead cell tissue debris breakdown particles from dying, rotting cells in their petri dishes. You must isolate that particle, alone and apart, from all of that other stuff to prove it is a virus and not just cellular debris garbage.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
No, it specifically means to remove that particle alone and apart from all of the other identical cellular garbage decay and genetically and chemically characterize it to prove it is a unique thing called a virus and not just more cellular garbage.
Isolation, what was it?
Isolation and purification.
Of the virus?
Yes.
Well, you isolate a virus by finding the virus which causes a disease. You purify a virus by making a lot of, just by purifying it so you get a pure virus.
Okay.
I don't understand what the issue.
They interchange the two, and I wasn't sure
I see.
If it was the same thing or if it was two totally different.
No, it depends on how they use it.
Okay. Can you explain the process of HIV isolation?
Well, didn't Dr. Gallo do that? He actually isolated it. So,
Yeah.
why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. I got other things to do.
A viral particle must fulfill defined physical and biological properties, including being a replication-competent intracellular parasite, meaning it results in identical copies of itself inside a host organism, and also capable of causing disease in a host, such as a human. However, viruses such as SARS-CoV-2 are nothing more than phantom constructs, existing only in imaginations and computer simulations. In this paradigm, cases of invented diseases like COVID-19 are nothing more than the detection of selected, tiny common genetic sequences and proteins purported, or rather pretended, to be viral. The existence of an actual virus is not required in this fallacy loop of circular reasoning, and thus, entire fictional pandemics can be built upon fictional digital creations and then falsely sustained through meaningless in vitro test tube molecular reactions.